So I just want to introduce the next speaker, Dr. Rebecca Sims from Cardiff. Rebecca is going to talk to us about profiling Alzheimer's disease for personalised medicine. Rebecca is a co-investigator at the UK Dementia Research Institute and is lead for the joint programme for neurodegenerative diseases. Thanks very much, Rebecca.
Hi, so thank you for the invitation to come present. So I'm going to take you back probably a little bit more basic to real basic science, not anything that's gone into clinical trials yet, and hopefully one day will. But I'm going to talk to you today about Alzheimer's disease and profiling that from the genetics. This is very genetic heavy, and I suppose I should say at the front of the talk, this isn't all about genetics and Alzheimer's. There are environmental factors, there's other things going on, but we specifically look at the genetics. So to start with, what is Alzheimer's disease? So there's obviously multiple definitions of Alzheimer's disease, which I've put on here. I'm not going to read them to you. But I wanted to really get across from this is that it's quite different and it can be quite heterogeneous. So actually, it's thought of as one homogeneous disease, but it's not. We get a lot of different symptom profiles. We get a lot of different outcomes, progression rates, etc. It's the man himself that defined the disease, and the first case study that he looked at, who also had psychosis within disease. So, bringing it right back, Alzheimer's is a form of dementia. So dementia is an umbrella term for various neurodegenerative diseases, of which Alzheimer's is the most common and accounts for 60 to 80%. However, as we're doing more post-mortem investigations, the amount of mixed dementia is becoming more apparent, especially in those with older age. And the genetic heritability of Alzheimer's disease specifically is between 56 to 100%, and I'll explain why there's such a wide variety in a moment. So firstly, hallmarks of Alzheimer's. So what do you have to be defined as Alzheimer's disease? So in the brain, you have to have the intracellular amyloid plaques, so senile plaques, and the extracellular neurofibrillary So this is amyloid beta protein, and well, among other things, but the majority is amyloid beta, and this is mainly tau protein. And essentially what you see in disease, before disease symptoms, which is part of the issue with defining those with dementia and Alzheimer's disease, is that you don't actually see symptom progress until there's a lot destroyed in the brain. There's been a lot of cell death, a lot of accumulation of the plaques and the tangles, and the only thing really that we see all the way through is your genetic predisposition, which is why we're so interested in SNPs and ApoE and other genes. So the genetic forms, you'll hear probably quite a lot about familial and sporadic Alzheimer's disease. So familial disease is almost always early onset. Well, you have causative mutations in specific genes, you will get disease. Sporadic's a bit more complicated. We do see early onset forms though. Not all early onset disease is familial. But more commonly, it's later onset after the age of 65. So the big hypothesis in Alzheimer's disease for many years has been the amyloid cascade hypothesis, which for familial disease fits very well. You have mutations in the APP gene or one of the presenilin genes, which are part of this amyloid cascade pathway, so how amyloid beta is broken down, which is the protein that's in the amyloid plaques, and how it aggregates together and causes plaques or is on the non-toxic pathway. ApoE, you've probably heard mentioned a lot, and I'll describe that a bit more in a minute. It's the biggest genetic risk factor across sporadic disease, and it's also linked to familial forms because it forms part of this pathway. It also does a lot of other things. So within ApoE, there's 2-point mutations that cause change in the protein, and essentially if you have the ApoE form, you're more likely to get disease. I've got another slide, actually. So based on that, if you've got 2 E4s, you normally get disease in your early 70s. E3s are later 70s. This is a sort of wild type, if you like, whereas the E2s are slightly protective. So if you've got 2 E2s, very rare. But if you've got E2 E2s, you're much less likely to get disease until a lot later. So obviously the fold change is huge compared to any other genetic variants we've got. But essentially, ApoE does a massive amount of things. This is what the mutations cause, the difference in the folding of the protein. But as you can see, there's massive different functions, and how you perform in those functions is dependent on this isoform in many cases. Okay, so I just wanted to give you a quick timeline of where we've been with Alzheimer's genetics. It's about the time I started my PhD. So before that, a lot of candidate gene studies, a lot of prior hypotheses of which genes were involved in disease. ApoE was identified in the early 90s. We started off with genome-wide pooling studies, which is essentially reduce costs, put all your samples together and estimate your allele frequencies. Over time, they have obviously expanded, and there are multiple genome-wide association studies that have been done. The most recent I'm going to talk to you about shortly, which was part of our JPND-funded project. And we're currently doing a mega meta-analysis, so it's trying to put everything from everybody internationally together, and also looking at doing an earlier onset GWAS, with the idea being that the earlier you get disease, the higher your genetic predisposition. So this is the most recent GWAS, so you can see we've got lots of genes, 92 now. This was over 100,000 clinical or proxy cases of AD, though, so you see we need massive sample numbers to get these findings, and the effect sizes are still pretty small, apart from ApoE, which is chopped off here. It's actually way up here somewhere. There's been a lot more with rare variants. That's common variants, where we see them quite commonly in the population, over 1% frequency. These are more rare, so we've also got hits in rarer genes. And this is sort of a schematic of pretty much everything that was just as the EADB paper was coming out. But what's interesting from all of this, and what's important from all of this, is loads of genes. It's really great, but actually they're not, and you can't read this, I'm afraid, but it's part of the EADB paper if anybody's actually interested, is they aren't separate in their function. A lot of these genes link together in biological pathways. They are doing similar functions, and we've got these other sort of key, this turns into this, these are the key pathways that come out of the research and the analysis we do on these variations. So a lot to do with immunity, a lot to do with protein trafficking, cholesterol metabolism. How we're using these now is within polygenic risk scores. So this is where we sort of link to the precision medicine potentially. So polygenic risk score essentially provides a measure of your disease risk, your cumulative risk due to your genetic architecture. Ideally, it needs to be linked with other environmental, phenotypic changes, but what we've been working on most recently is defining the genetic element of this. And essentially what it says is that if you're a control, you've got a lower cumulative risk than a case. Most of us are going to be in the middle. We're not going to have extreme risk in one direction or the other, but there are people that are on the lower and the higher extremes, and those are the most informative people for us, and that's what our research is focusing on at the moment. So this paper was published, I think, about 8 years ago now. It's, 2015. So just with the genetics alone, including ApoE is the most significant risk factor, sex and age as well as the biggest environmental factors, we could get up to accuracy of about 78%, which meant 78% of the time we were right if they were a case or a control when we estimated. And this is sort of how you can see with the different polygenic ranges. So if you're very low risk or very high risk, this is where we sort of were getting our accuracies across. So the lower risk you are, the more accurate we are. The higher risk you are, the more accurate we are. So this was then funded as part of an Innovate UK grant, and there is now a genetic risk score available based on this work through Cytox. So essentially you can send them a sample and they will screen your blood based on the work that we've done. But what we're doing now is trying to improve this. So it's great, we get accuracy of 78% of the time, but that's not clinically useful. We can't define who's most at risk well enough to put that into sort of clinical trials. So what we're doing at the moment is trying to improve this, looking at new genes, looking at pathway-specific risks. So we see that some people have much more genetic risk in the immune system, for example. Others have much more genetic risk in the cholesterol. And we're currently now trying to, with a PhD student in Bristol, Peter, trying to integrate this with phenotype data, so data we have on them based on activities of daily living, also their symptom profiles, to see if we can really build up a true risk profile, not just based on the genetics. But what we're really interested in for the sort of molecular science side of things is participant selection. So trying to identify the right people and the right samples to work on based on the idea that these would be more susceptible to certain approaches than others. So there's 3 key projects that the DRI in Cardiff is working on. So ITMAR, which is Julie Williams, I'm going to describe them all slightly. Biomarker Factory, which is a collaboration across Cardiff and UCL. And Milton Mills and Imogen, and Tom's just moved to Bath, so he's taken that work with him. So getting new genes, extremes of disease. I mentioned early onset disease, so there are sporadic forms of early onset disease where you get disease early, but you don't have causative risk mutations. So the idea is that we have high impact variants in these people, and we've seen from our research so far that people at a younger age have a higher effect size of some of the genetic risk factors. So we're currently working on a genome-wide association study of early onset Alzheimer's disease. Lots of different groups together. It takes a lot of time. It's interesting collaboration. You've got sort of 400 scientists trying to work together. It can be a little bit ahead of a challenge, but we are getting there. We've got about 12,000 non-familial early onset cases that we've identified and over 60,000 controls that we're putting together. So hopefully this is going to be published at least on bioRxiv or something by the end of this year, beginning of next. There should be a title in here, sorry, it's gone. Another area that I'm particularly interested in is the hidden genome. So everything we've done so far has been based on microarray analysis and short-read next-generation sequencing. There's actually a huge amount of the genome that isn't picked up by those methodologies. It's something that's often called the hidden genome or the dark genome, and that's where long-read sequencing is coming into effect. We see a lot of genetic risk in the complement pathway, which is a part of the immune system, and a lot of those genes aren't covered by the arrays and the short-read sequencing techniques that we currently have. So I'm really interested in trying to use long-read sequencing to try and look at some of these genes and look at whether there's something more significant with regards to a structural variation, something protein changing that we can look for. So ITMA has been going about 3 years now. It's essentially a platform to look at induced pluripotent stem cells from people with high risk for Alzheimer's or low risk for Alzheimer's. So it's basically taking PBMCs, transforming them into iPS cells and then differentiating them primarily into neurons and microglia, because that's the sort of cell types of most interest from the work that we've been doing. So the team are doing really well, and we've got a few lines now established that are going to be then available for others to use through either Dementia Research Institute or the HIPSCI bank. So the Biomarker Factory was published last year, actually. This is a collaboration between Cardiff and UCL. So UCL got a lot of experience in blood-based biomarkers for Alzheimer's disease. So essentially what we did was take Alzheimer's cases that we had genetic profiles for and screen controls and measured plasma concentrations of various amyloid Alzheimer's-specific markers, so A-beta 40, 42, tau, and looked at doing a genetic risk, basically a genetic association study for the biomarkers, and we identified 2 genes that were linked specifically to amyloid and p-tau, those were. So this was a genome-wide association study. And then multimodal imaging, so using those at high and low risk for Alzheimer's disease and imaging them with MRI at 3T and 7T, and this is Tom's work, who's now moved to Bath. So looking at the differences in these people on these scans. And the idea is essentially that we want to try and integrate this data into a score so that we can get better at predicting who is going to be getting disease. So just to say, the Cardiff DRI, this is basically what we're all doing. So there's sort of different arms of research, all stemming from the genetics, but essentially looking at specific mutations, functional mutations, and hub loci. So there's a couple of the genes we've identified as transcription factors. That, so looking at those in human and animal models. Also then taking the polygenic risk extremes, either whole risk for the whole of disease or pathway specific, and then looking at hopefully getting to the point of clinical trials once we improve the scores, but also taking them into human and animal models, and then obviously measuring outputs, so hopefully identifying new mechanisms and new therapies going forwards. So thank you all for your time. I just wanted to highlight, we've got loads of collaborations, but I really want to highlight the team in Cardiff that do pretty much all of this work, collecting the samples, analysing the data, and obviously the PIs of the projects.